Chemists at MIT have developed a novel way to synthesize Himastatin, a natural compound that has shown potential as an antibiotic. Using their new synthesis, the researchers were able not only to produce Himastatin but also to generate variants of the molecule, some of which also showed antimicrobial activity. They also discovered that the compound appears to kill bacteria by disrupting their cell membranes. The researchers now hope to design other molecules that could have even stronger antibiotic activity. Himastatin, which is produced by a species of soil bacteria, was first discovered in the 1990s. In animal studies, it was found to have anti-cancer activity, but the record doses had toxic side effects. The compound also showed potential antimicrobial activity, but that potential hasn't been explored in detail. Himastatin is a complex molecule that consists of two identical subunits known as monomers that join together to form a dimer. The two subunits are hooked together by a bond that connects a 6-carbon ring in one of the monomers to the identical ring in the other monomer. This carbon-carbon bond is critical for the molecule's antimicrobial activity. In previous efforts to synthesize Himastatin, researchers have tried to make that bond first using two simple subunits and then added more complex chemical groups onto the monomers. The MIT team took a different approach inspired by the way this reaction is performed in bacteria that produce Himastatin. Those bacteria have an enzyme that can join the two monomers as the very last step of the synthesis, by turning each of the carbon atoms that need to be joined together into highly reactive radicals. To mimic that process, the researchers first built complex monomers from amino acid building blocks, helped by a rapid peptide synthesis technology. The researchers then used a new dimerization strategy to connect two complex molecules together. This new dimerization is based on the oxidation of aniline to form carbon radicals in each molecule. These radicals can react to form the carbon-carbon bond that hooks the two monomers together. Using this approach, the researchers can create dimers that contain different types of subunits in addition to naturally occurring Himastatin dimers. One of the variants that the researchers created has a fluorescent tag, which they used to visualize how Himastatin interacts with bacterial cells. Using these fluorescent probes, the researchers found that the drug accumulates in the bacterial cell membranes. This led them to hypothesize that it works by disrupting the cell membrane, which is also a mechanism used by at least one FDA-approved antibiotic, daptomycin. The researchers also designed several other Himastatin variants by swapping in different atoms in specific parts of the molecule and tested their antimicrobial activity against 6 bacterial strains. They found that some of these compounds had strong activity, but only if they included one naturally occurring monomer along with one that was different. The researchers now plan to design more variants that they hope might have more potent antibiotic activity.